My name is Ava Laszlo, and today I will be presenting to you a presentation created by myself, Dalen Woolnough, and David Zanatta. My title is Predicting Presence of Freshwater Mussel Populations Using Habitat Parameters in 2 Reservoir Lakes. The Tittabawassee River watershed is approximately 4,000 square kilometers located in central Michigan. Within the watershed is a series of 4 reservoir lakes. These lakes are Secord Lake, Smallwood Lake, Wixom Lake, and Sanford Lake. These lakes were created in 1925 through the process of building dams in 1923 through 1925. May 17th through May 19th, 2020, a record rainfall event occurred in Midland and Gladwin counties. This record rainfall event, combined with pre-existing dam infrastructure issues, led to damage of all 4 reservoir dams and the surrounding areas, flooding of the 4 lakes region, consequent water loss when reservoirs were drained from dam failure or drawn down to inspect damages, and water loss affected aquatic animals, including freshwater mussels within family Unionidae. Freshwater mussels provide many ecosystem services, including biofiltration, nutrient cycling, and structural habitat. The Tittabawassee River watershed is lacking in Unionid mussel survey data in the 4 Lakes region, with one exception, a study by Hunter Dan. However, this study was tributary focused and not lake focused. Our overall study priorities were to perform freshwater mussel surveys in Secord and Smallwood lakes in 2021 and Wixom and Sanford lakes in 2022 through 2023. Our 2021 Secord and Smallwood study goals were to 1. Determine presence and absence of Unionids, including listed Unionid species. 2. Examine Unionid habitat and create a mussel presence predictor model based on our findings. And lastly, to identify threats to Unionids. We used ArcGIS digital mapping software to generate and identify random sites within the lakes at least 50 meters apart. We manually selected non-random targeted sites, including tributary sites, historical sites, and sites that were adjacent to the dams. Once at a survey site, we would assess the site and perform surveys using snorkel and scuba if safe and accessible. We surveyed for both live and shell Unionids, presence of invasive species, and habitat characteristics. To document Unionid habitat, we used the Wentworth sediment scale to estimate composition of bottom habitat. The scale is a way to visually assess sediment type using classes of grain size. So on the figure on the right, we have smaller grains at the bottom of the scale, including clay, silt, and sand, and larger grains at the top of the scale, including pebbles, cobbles, and boulders. For our results, we were able to sample 59 survey sites and 108 person-hours. We documented 13 species total represented by live or shell, including 448 live individuals of 6 different species. We also collected 1,457 valves, or half shells. 94% of the Unionids we collected alive were giant floater Pyganodontodon grandis. Now, what do we do with this mussel survey data? We decided to perform a CART analysis. CART stands for Classification and Regression Tree. It is a type of decision tree used to predict an outcome. So first, you start with your survey data. In our case, our habitat characteristic predictor variables were our Wentworth scale variables and a few other variables, including mean site depth, and categorical variables such as if a site had Unionids present or Unionids absent. The model building process selects variables of high importance to predict outcomes. So in this example decision tree, we have sites that have sand less than 2.5%. The model predicts Unionids will be present there. Sand with greater than or equal to 2.5% composition at the site, the model predicts Unionids will be absent there. So for our CART analysis, we first extracted lake site data. So we used 43 of the original 59 survey sites. We performed k-fold cross-validation and grew the tree using the rpart package in R, and then we pruned the tree. So how we read a decision tree with cart analysis is we start at the top and we work our way down. So in this example, we have clay greater than or equal to 2.5%. The model predicts that unionids will be present. And this is our first leaf node. We can also look at the accuracy of each node. So for our first leaf node, n is equal to 12. This is the number of sites that the model used to create this leaf. The model is 100% accurate in this leaf because we have 12 unionid present sites that were used to create this model, and 0 of the sites the model used were unionid absent. We then made a confusion matrix to estimate the overall model accuracy. So we have our model predicted values against the actual values of the sites. Our model correctly predicted 29 sites as true positives and 9 sites as true negatives. Our model incorrectly predicted 2 sites as false positives and 3 sites as false negatives. When trying to find rare species, the worst case is a false negative. This is when the model predicts that unionids will be absent when they are actually present. A pruned tree predicted 5 out of 43 sites incorrectly, so our accuracy value is 38/43, or roughly 88%. A different measure of classification model performance other than accuracy, which has been suggested in the literature to be a more robust model performance measure, is the ROC, or Receiver Operating Characteristic Curve. This is associated with an AUC value, or area under the ROC curve, which has a value between 0 to 1. But before I talk about ROC curves anymore, I wanted to talk about the ROC space. And so on our x-axis, we have the false positive rate, also known as 1 minus specificity. And on the y-axis, we have our true positive rate, also known as sensitivity. The y-axis and the x-axis both range from 0 to 1. At the point coordinates 0, 0, the model will always predict negative. At 0, 1, this is perfect model accuracy. At 1, 0, the model is perfectly inaccurate. And at 1, 1, the model always predicts positive. This is an example of an AUC value of 1. This is equivalent to a perfect model. In this example, this has an AUC value of 0.5 and is equivalent to a random model. AUC values between 0.5 and 0.7 are poor, indicating that the model is either random or only slightly more sensitive than the rate for false positives. This is our ROC curve, and our AUC value is 0.939. Models with values of above 0.9 are considered, or equal to 0.9, are considered to be good and have a high rate of sensitivity. Now let's talk about threats. Greater than 90% of sites in both Secord and Smallwood lakes had live Dracena polymorpha, also known as zebra mussels. Zebra mussels were more intense at sites in Secord Lake, and unionid fouling was more frequent and more intense in Secord Lake. So we tried incorporating our zebra mussel and site intensities into our cart model. Our first model used unionid present and unionid absent variables, as well as low intensity, medium intensity, and high intensity zebra mussels at each site. For this first model, we have a mean multi-class AUC value of 0.7. Our next model, we combined low and medium and left the high sites alone. And this is a AUC value of 0.71. In our last model, we left low by itself, and we combined medium and high intensities, and we got a mean multi-class AUC value of 0.8. So let's go back to our original cart without the zebra mussel information, with the AUC of 0.939. So leaving the zebra mussel intensities at the site information out of our CART model, we then created a series of bar charts that depict zebra mussel site intensity and zebra mussel fouling intensity on the x-axis and the percent density category on the y-axis. And so for our colors, for black, we have Na, absent is blue, low intensity green, medium intensity orange, and high intensity red. So let's talk about our first leaf, where the model predicts Unionids will be present if clay is greater than or equal to 2.5% at the site. Within this leaf, our N is equal to 12, so 12 sites were used to create this leaf. We notice that our site intensity of zebra mussels is the least in this leaf out of the rest of the bar charts, and that our zebra mussel fouling intensity is also the least in this particular leaf node out of all of the bar charts. Few studies have been done with unionids in reservoirs. Our methods could be used as baseline for future studies. Our original model without zebra mussel information is 88% accurate with an AUC of 0.939. Our other models suggest that zebra mussel presence and intensity are not necessarily predictors of unionid presence and absence. We noted often heavy biofouling by zebra mussel on unionids and hard surfaces. Depolymorpha zebra mussels compete with unionids for food, and fouling affects locomotion and feeding. Fouling also can cause shell deformities and even mortality. Our model suggests that clay may inhibit zebra mussels both at a site and fouled on unionids, as the texture of unconsolidated sediments may prevent settling and attachment of larvae, and unionids are able to burrow in soft sediments to dislodge zebra mussels and induce mortality and encrusted zebra mussels due to low dissolved oxygen in the sediments. I would like to thank all of our field and lab help with this particular study, everyone involved with logistics and feedback, and our funding source, the Four Lakes Task Force. If you have questions or comments, you can ask them now or feel free to contact myself, Dalen Woolnough, or Dave Zanatta through email or social media. Thank you.